Hello, I'm Rubab, your audio navigator, and I'm here to guide you about how to prepare for GATE Life Sciences. As you know, it has one compulsory section that is chemistry, and rest of them are optional. You can choose any two sections from the optional papers according to your field and interest. So here I will tell you about how to prepare for biochemistry if you are choosing one as an optional paper. If you are studying biochemistry, then you will obviously choose biochemistry as one of the optional paper. But if you are studying microbiology or food technology, then also you can choose it. It will be a nice combination. So let's start with the first section of biochemistry, that is about biomolecules. As I've already told that for preparing GATE exam, you need to first of all clear your basic concepts about each and everything you are preparing. So keep that in mind always. This section is very vast as it contains all kinds of biomolecules, their structure, their functions. A lot of basic concept-based questions could come from here regarding the structure or the function. Mainly 1 to 2 marks. So start with the organization of life. So for that you should know what are the different levels of the organization, like from atoms to molecules to biomolecular complex to organelle to cell to tissue, then organ. They form the organ system, then the organism, population, community, ecosystem, biome, and then the biosphere. Each level is very important for the organization. So you should have the basic idea about all of these. How do they take place? Like how the atoms combine to make a molecule. Then what are the different bonds which are involved, like electrostatic bonds, hydrogen bonds, van der Waals bonds, etc. Or the covalent bonds. Then what are the dimensions in which all these fall? For example, from the atoms to molecules to macromolecules to assemblies to cells. So what are the different magnitudes? Then what are the different energy levels of different molecules? So these are the very basic things which you need to know. After that, you need to study a very important part of life, that is water. So what are the important properties of water which makes it very important for all molecular interactions in biological systems? It is that water is a polar molecule, and the other one is the cohesiveness of water. Then what are the other properties? It solvates polar molecules and hence weakens ionic and hydrogen bonds. So just you should know how it happens and how does it influence the non-polar groups which tend to associate in water by hydrophobic attractions. Then after covering the importance of water, you need to go through the structure and function of different biomolecules. Let's start with the protein structure and function. So you should know what are the important functions which proteins have. So what role do they have in enzyme catalysis, transport and storage, coordinated motion, mechanical support, immune protection, generation and transmission of nerve impulses, control of growth and differentiation. So you should know how proteins perform all these functions in detail. Then coming to the structure of proteins. Proteins are built from a repertoire of 20 amino acids. So you should know about all these amino acids, their structures, the functional groups which they have, and their pK values. And of course the one letter symbol which they depict. After that, you should study about the peptide bond, the linkage through which they are linked. What are the properties of a peptide bond? Then what are the other linkages which are found in protein molecule? For example, the disulfide bond. Then what are the secondary structures in which the polypeptide chains can fold and form regular structures? For example, the alpha helix and the beta pleated sheet. So you also should know about the dimensions related to these structures. For example, what is the distance between two alpha carbon atoms? How many amino acid residues are there in per turn? What are the hairpin turns? So you need to go through about the properties of each of these structures and also where they are predominantly found in which protein molecules with examples. Then what are the other aspects on which the structure of a protein molecule depends? That is, the proteins are rich in hydrogen bonding potentiality. Then the water soluble proteins fold into compact structures with non-polar cores. Then what are the four basic levels of structure in protein architecture? Then you should know how the structure of a protein is dependent upon the amino acid sequence. So you need to study this with examples. Then how the specific binding and transmission of structural changes are the responsible factors for the protein action. Then after that, you should know how protein folding happens. There are four basic structures of protein like the primary, secondary, tertiary and quaternary. So you should know how the proteins fold by progressive stabilization of intermediates rather than by random search. And how the molten globules containing native secondary structure are formed early in the folding process. Then what are Ramachandran plots? How do it helps in predicting the protein structure? Then you should know that the amino acid residues have different propensities for forming alpha helices or beta sheets or beta turns. So you should know what are the different amino acids which promote alpha helix or beta sheet or beta turn. While studying the folding process, partially folded intermediates can be detected, trapped and characterized by different processes. For example, by rapid kinetic studies or by trapping of disulfide bonded intermediates or by pulse label NMR studies or the synthesis of folding kernels or site-specific mutagenesis or the protein design. And so go through different examples and should know how the folding process takes place. For example, in lysozyme it folds along the multiple parallel pathways, while in trypsin inhibitor native disulfide bond intermediates predominates the folding process. Then how the folding process catalyzed in vivo, for example by the isomerases and chaperone proteins. So you should know about these and should also know how do they help in folding. And you should know that many proteins are marginally stable. So what is the biological advantage of this? For example, protein flexibility is at the heart of protein action. Then the nascent proteins must not be allowed to fall into deep traps en route to the native state. Then the partial unfolding enables protein to be transported across membrane barriers. Then the proteins are constructed so that they can readily be taken apart. So go through these points in detail. After covering these points, you should also know that how the protein sequencing is done. There are traditional orthodox methods and there are new methods also. So go through them. How the recombinant DNA technology has revolutionized protein sequencing. And you should also know that how the amino acid sequences are helpful for different purposes. For example, the sequence of a protein of interest can be compared with all the other known sequences to know that to which family does that protein belongs. Then it can also be helpful in giving the information about evolutionary pathways. Then it can also be helpful in telling us about the presence of internal repeats. It can also be helpful in the determination and destination of proteins and control their processing. And the sequence data provides a basis for preparing antibodies specific for a protein of interest. And it could also be helpful in preparing DNA probes. So you need to go through all these points in detail. And this portion is very important for the exam point of view. You should also know about the different tools and methods which are used for predicting the secondary, tertiary and quaternary structures of protein. After amino acids and proteins, you need to go through carbohydrates. So for that, first of all, you should know what are the main roles of carbohydrates. For example, like they work as energy stores, fuels and metabolic intermediates. Carbohydrate units on cell surfaces play key roles in cell recognition processes. Then what is the structure of a carbohydrate? Then what are monosaccharides and what is the structure of monosaccharide? Then what are the D and L forms of monosaccharides? They represent the absolute configuration. Then the pentoses and hexoses cyclize to form furanose and pyranose rings. So you should know how does it happen. Then what is the conformation of pyranose and furanose rings? Then what are glycosidic bonds? They are the bonds which join carbohydrates to alcohols and amines. Then what are phosphorylated sugars and what are their roles? For example, mostly they play key intermediates in energy generation and biosynthesis. Then after that, what are disaccharides? What are the common disaccharides known and their structures? Then what is lactose intolerance? Then what are glycogen, starch and dextrin? These are the mobilizable stores of glucose. Then what is cellulose and what is the basic structure of cellulose? Then you need to know the different structures of commonly known carbohydrates, for example, like glucose aminoglycans, proteoglycans. Then what are oligosaccharides and what are their major roles? For example, sometimes they are attached to integral membrane proteins and many secreted proteins. So you should know about the examples of this. Then after that, what are lectins and what are their roles? Then what are the roles of carbohydrate in cell adhesion? What are the carbohydrates which are involved in this and their structure and the mechanism also. After going through the structure and function of carbohydrates, you need to do the same for lipids. You should know what are lipids and what are the general characteristics of lipids. Then what are the different kinds of lipids? What are simple lipids like fatty acids? These are the esters of fatty acids. They could be saturated or unsaturated. And the unsaturated could be mono or polyunsaturated. Then what are trans fats? Omega fatty acids. Then what are trans fatty acid, and then what are triglycerides? You should know about the structure of all these. And then what are waxes? Then what are compound lipids like phospholipids, phosphoglycerides, phosphoinositides, glycolipids, etc. We should know about the structure and function of all these. And after that, what are derived lipids? They could be steroids, terpenes, or carotenoids. First of all, study about the steroids. Then what is the structure of cholesterol? Then there are a few general terms which are used with regards to the lipids, so you should know about them. For example, essential fatty acids, non-essential fatty acids, unsaturated and saturated fatty acids. So lipids could be divided into three categories like phospholipids, glycolipids, or the polyphenol compounds. So you need to know the examples of all these and plus the functions and structure of these lipids. Then what are the biological functions of lipids? For example, in membranes, the glycerophospholipids are the main structural component of biological membranes. So you should know about the structure and function. Then they have role in energy storage, signaling, etc. Plus they could be divided into fat soluble and fat insoluble lipids. Sorry, the vitamins are divided into fat soluble and fat insoluble groups. Fat soluble vitamins that are vitamin D, E, and K. These are isoprene based lipids and are essential nutrients stored in the liver and fatty tissues with a diverse range of functions. Likewise, there are various functions of lipids. Go through them.